Hi everyone, welcome back to my channel Synaptic Signal, and today I brought the topic genetics. So genetic is most fascinating branches of biology, and this is one of the important lessons which you need to cover in the unit heredity. So students, you might get confused like some of the terminologies, the new words might confuse you, and you might be struggling with genetics. Is very hard to study topic. So I would rather skip it. But don't skip it. Just the new words that are you are struggling with. So let's clear it out, and let me first take you back in time in mid 1800s. So a question arises, who is Gregor Mendel? Let us know who is he. Gregor Mendel, yes, he is called the father of genetics. So what he did was he studied pea plants. So what he did was he studied pea plants, and he also began experimenting with the pea plants to study how traits are inherited. So he discovered principles of heredity. For an example, how traits are passed from parents to the offsprings. Later on, for previously his findings were not like overlooked or something like that, and later on, people compile it, and now you have to study it as the Mendel's law. So, which is the foundation of modern genetics? Let me tell you that. After we became familiar with Mendel, now this portion, as you can see it in the slide, the question might arise also, why pea plant? And why pea plant were the brilliant choice for Mendel experiment? As you might be easily saying, it is easy to grow, it is easily found, or something like that. But if you have to write, will you be writing in just one sentence? No, it's a big no. So please, this is the summarized points which I have written it from your curriculum book. First point is natural self-pollination. That means being a bisexual flower, naturally it is self-pollinating plant, and also it ensures, which ensures now genetic consistency over here. And next thing is, what we can do is we can researchers, not only we researchers can easily intervene, cross pollinate them when needed. So it is a control process where they can easily cross pollinate whenever it is needed, and it has a short life cycle. So that means its multiple generation can be studied with the rapid observations. So next point is the quite important point, where clear trait contrast. That means pea plants, they display many distinguishable traits, as you all can see in the screen also, like tall versus short, which is quite perfect for tracking inheritance. And next thing is, for the large sample sizes for experiments, what do we need? We need uh, many seeds, isn't it? So it produces high seed production, so that for the experiment the sample size is large. So in this slide you can see Mendel's 7 traits of inheritance. So you might be confused in the book that it might be giving you what are those 7 traits. So these are the 7 traits, and at the side you could see dominant and recessive one. So dominant one are that gets expressed. Recessive one, that means in presence of dominant traits they cannot express itself, they get hidden. So for an example, for the first trait, which is flower color, purple is the dominant one which get expressed, and recessive one is the white one which doesn't get expressed. Same goes for this sheet shape, that is round is the dominant, wrinkles is the recessive one, sorry. And you might be confused, what is pod? Pod is the seed vessel. So inflated, see, pod shape, inflated one are the dominant ones, constricted ones are the recessive ones. So you might be seeing it in the book also, and I'll be mentioning also. You might be wondering what are alleles. So if you're confused what is alleles, there are different versions of gene. For an example, let us imagine gene like ice cream category and the different flavors, such as what do you like, the ice cream flavors. Most of the people they like chocolate, isn't it? Mostly, or mostly people like vanilla. So chocolate, vanilla, strawberry. Let us imagine these flavors are alleles. Genes as the ice cream flavor category, and different flavors are the alleles. So they are all variations of the same thing, but each gives different outcomes. For an example, each flavor of the ice cream gives different taste. So we can summarize alleles as the different versions of gene, as I've told you before, and the combination you inherited from your parents, which decides which trait you will be showing. And here on the screen, you could see homozygous and heterozygous alleles. So I've supposed capital A and A. These both alleles are identical. That means the individual is homozygous for this gene. And if the capital A and the small a, if both alleles are different, the individual is heterozygous for this gene. Now, this slide shows you Mendel's genetic experiment method. So, I've summarized the Mendel's genetic experiment method starting from selection of pure lines. So, what he did was he chose pure tall and pure dwarf pea plants to ensure consistent traits. And then Mendel did what he did was he cross pollinated the tall and dwarf plant. Now, what he did, he took the observation of this first generation, where he could see every, the tall trait dominated the dwarf one. And then what he did was he self pollinated the F1 generation. After self pollination, the second generation was the tall and dwarf both get expressed. So, reappearing the recessive dwarf trait again. Summarizing, what we can say is Mendel found out when the we cross plants with contrasting traits, that is, tall versus dwarf, the dominant trait appeared in the first generation F1 generation, while the recessive trait they reappeared in the second generation, making the phenotypic ratio 3 is to 1 and genotypic ratio 1 is to 2 is to 1. So, as I was talking before, the findings of the Mendel experiments they were compiled and which were the foundations of modern genetics nowadays is the Mendel's law. It all starts from the law of uh, dominance. So let us focus on this slide, the law of dominance. So what is it? So understanding how dominant traits manifest in an organisms. So let's explain this particular line. So when two pure individuals of contrasting traits are crossed, only one trait appears in the first generation, which I've already explained in the uh, Mendel's experiment also. For an example, the purple flower when crossed with white flower, uh, the first generation are all purple flower. Only purple color will be expressed as it is the dominant one. And here down, I've given you a figure also where which you can draw easily draw it in the exam and explain this topic. So What you will do is during exams, what you do is most of the students, what they do is they keep on like explaining, writing, and even if they knew how to explain it with the figures or charts or tables or diagrams, they avoid it. So it is better when you draw figures and explain it a little bit, and not little bit, I'm not saying little bit only, but explain it nicely and also with the figures. So over here in the figure, what happens in the first generation, when this tall plant it crosses with the dwarf plant, only tall trait is expressed. The tall one is the dominant trait and the dwarf is the recessive trait. Now I think we're a little bit clear about the law of dominance. Now let's get back to the law of segregations. So in this law of segregation, it explains how alleles separates during gamete formation. That means each individual they carry two alleles for a trait, but only one allele is expressed. For an example, a genotype, capital T and small t. It is for what? Tall. It produces gametes either capital T or small t. This explains why the trait appears in the second generation, which is F2 generations. So the recessive trait they reappear in the second generation. So this third law, as you can see, law of independent assortment. So let me tell you to highlight on the word independent. That means the inheritance of one trait does not affect the inheritance of another trait. For an example, the plant height does not affect the flower color. As I've given example also. So as you all can see on the screen, so its main idea is alleles of different gene assort independently during gamete formation. So this law is held to be true only when the genes they are located in the different chromosomes. So I have included a monohybrid cross between tall plant and dwarf plant, which is up to the F3 generation, which I've kept from our curriculum book, science book, which you can use it as your reference. You can pause it and just see. I have already explained this, and also I would like to suggest you to make a tabulated form of monohybrid cross and dihybrid cross. And let me know if you want a tabulated form or something in the comment section below. So it was a quick walk in the Gregor Mendel's work.